I'm very happy to welcome everyone here for the second session of the National Webinar Series in Bioinformatics and Computational Biology on behalf of SASTRA and COMBIX. And COMBIX is a student association of the Department of Bioinformatics in SASTRA. We have this kind of webinars and conferences, workshops, etc. Apart from the academic setup, so that the students would be able to know what is happening in the industry as well as in the academia around the world. What are the recent trends? So, with that, a very small introduction. Without much further ado, I'd request Mr. Taranna to introduce the speaker, and then we'll go ahead with this webinar. Taranna, can you take over?
Yes, sir.
Please?
Yes, sir. Thank you. Good evening, everybody. I hope I'm audible. Thank you for joining today's webinar. It is my absolute privilege to introduce our esteemed speaker, Dr. Khader, a distinguished leader in AI-driven drug discovery and advanced healthcare. He currently serves as the Executive Director and Global Head of Data Science at Sanofi, where he leads pioneering initiatives in biomedical research. Dr. Khader holds a PhD in computational biology from the National Center for Biological Sciences and completed his postdoctoral training in computational genomics and precision medicine at the Mayo Clinic. In addition to his PhD, Dr. Khader has pursued specialization programs in public health from Imperial College London, health information technology at Harvard, machine learning for big data at MIT, and entrepreneurship at Cambridge Judge Business School, equipping him with a multidisciplinary edge in healthcare innovation. With extensive expertise in bioinformatics, big data, and biomedical data science, Dr. Khader has made groundbreaking contributions across pharma, biotech, medtech, digital health, precision medicine, genomics, and drug development. His professional journey includes leadership roles at AstraZeneca, Philips, and Mayo Clinic, where he has been instrumental in advancing data-driven healthcare solutions. Recognized globally for his contributions, Dr. Khader has been named one of the 100 leading pioneers of AI drug development by Forbes and Deep Knowledge Ventures, and one of the 16 data scientists making vital breakthroughs in healthcare by Pocketblock. Dr. Khader, we truly appreciate your time and expertise today. It is an absolute honor to have you with us, sir.
Thank you. That was quite a long introduction, but I really appreciate. But before I get to the talk, I want to thank Suma for consistently reaching out and making sure that I become part of this this conversation, more like a conversation, but she was in touch probably since more than a year or so. Last year we tried to connect, but due to the time issue or something, we couldn't make it happen. But here we go. This shows that persistence is very important, and I'm really glad. I'm really glad that I think it's a Saturday evening, right? And I would assume we have a lot of students joining. So really, the honor is really mine to kind of share my version of the story, right? Really what we could do and what you all, each and every one of you, could do in pursuing studies or research in this area of of where we in a way combine biology and medicine and technology, and trying to save patient lives or improve lives of each and every one of us, because in one way or the other we'll all become patients, we'll all need access to medicines, access to healthcare, so we all will be part of that. So I'm going to switch to this mode. I hope everybody can see me, everybody can hear me. That's good?
Yes.
Excellent. And this is the view I will have till I switch it, but this is okay. So what we're going to do today is, before I start, there is no conflict of interest here. So the way that, this is an academic style talk where I spent a considerable amount of my talk today to talk about some of the way that artificial intelligence, or more or less like I like to call it, machine intelligence, is now changing the paradigm for each and every one of us. Because I saw that there is an application running here called Tactic to transcribe, just as an example. So AI is within each and every one of our lives. It's there, it's embedded. And then when we think about bioinformatics, like in the bioinformatics for just like you use mathematics, statistics, or other types of data visualization, AI approach is at the core of it as well. So my idea is really to tell you, not just my journey, but then again, what is happening in that field and why it is important to kind of bringing elements of AI into research problems that you are all looking at. And if you just before I start, just if you take my career, the first half is where I spend a lot of time, building large-scale AI systems for hospitals and health systems, and then over the last 8 years or so, I've been applying this into pharmaceutical development. This is drug discovery, and so kind of we go through a few examples in both healthcare as well as this as well. All right. So with that, we are in an interesting phase of humanity, right? Like, so there is a significant population growth, but at the same time, the disease burden is increasing, and we also have emerging outbreaks. And this is a Statista slide. It's probably from 2017 or 18, where they were projecting that by 2050, the drugs, the deaths from drug-resistant infections will be one of the major reasons for deaths, right? Like by 2050. And then, I used this one in a TEDx talk I gave in, if I remember correctly, 2018. And then many people were asking me, like, because at that time, infectious disease, we hear about some infectious disease and all, but we were not sure, none of us have experienced the kind of things we're talking about. And then what happened in 2019? COVID happened, right? Like, and we were not ready for that. But in one way or the other, the entire world came together, and we are out of that phase now. And is that it? We won't get another infectious disease. We don't know, right? Like, so it's an important part to kind of think about, and then at the same time, we also have emerging microbes, emerging infectious disease. It could be more and more virulent strains are being evolved as well. So this is something that I will keep in my deck for decades to come. But then just to kind of show that where things are going by 2050. One is a bit of a prediction, but the other one is a statistical estimate, and we will see how things work. But then, how do we combat? Whether this is an infectious disease, whether it's a communication, sort of lifestyle-based diseases. How do we do that? Our primary way is to look at data, and then, for that matter, biomedical data is rich, complex, digital, and natively connected. What do I mean by that? If you study one, if you're interested in studying one gene, you can definitely, you can clone it. You can potentially develop an inhibitor to attack it, but really that will only help you to study that gene, right? Like, but the biology is very, very complex. Once you try to turn off something here, you will have, let me just see if I can get there. Not that one. So once you try to turn, this gene, you'll probably have impact on the rest of the networks. And then when you try to interfere between a protein and DNA interaction, you could have other issues in downstream genes. And then that this classic theme in biomedicine that we all now realizing that no disease is an island, right? Like a patient could have diabetes, but then the genes that are involved in diabetes could also be involved in hypertension, could be also involved in atherosclerosis, and then also cardiovascular diseases, so on and so forth. But then here the idea then would be, can we develop therapeutics, in an optimal way? And that's one of the core things that we try to do as well. But then there is a bit of a challenge, right? Like, so in a typical health hospital system, what we do is we basically measure, we take a vial of blood, we measure, different parameters. We take blood pressure, we look at, CT, angiogram, and other types of echo and other types of measurements. But then there is a lot of things happening in other levels, right? Like, your exposome, the quality of the air, right? Like, that matters depending on whether you're from Delhi or Chennai or from another village in Tamil Nadu or Kerala. So the air quality there is considerable difference in that, and that have an impact on the disease aspect. And then the genomics, the genetics aspects, the transcriptomic, the RNA level. So even if you have a mutation, it doesn't mean that a patient will get a disease. So there are other mechanisms that goes between that. For example, like nonsense mediated decay and others that could potentially help the patient, but at the same time, sometimes the mutations are so strong could lead to a disease. And then again, we now have technologies to measure genome at scale. You can sequence a person and interpret that thanks to bioinformatics. You can do this, more or less in a day and at scale actually. The transcriptome is again another one, so we can do single cell. You can do what you call as a bulk transcriptome, where you look at the entire transcriptome from a tissue. But then nowadays you have another technique called single cell, where you can not only just do transcriptome, but in a cellular level. So you know what type of cells are involved in certain diseases. So both of them you can do in a pretty high depth, high coverage. But we're slowly catching up with proteomics. We could do maybe around 10,000 proteomic protein measurements at a time, and other things like the connectome, physiome, so on and so forth. So there are different ways. So one of the ways that you'll see in my research that we primarily do is try to connect. What can we predict a health outcome by measuring genetic data? Can this be predictive of some of the clinical aspects? So that's one of the things that I'm very much interested in. So now you have all of these technologies, right? Like you have internet, you have mobile, you have artificial intelligence, but the reality is that while the technology adoption is increasing, but the cost of the care is not decreasing. And whether it's in the US or in India or most of the places, the cost is actually extremely increased. And then how do we then one of the way that we could potentially address this is by looking into data. What's really happening? Like, what are the drivers of the cost? What are the drivers of the better outcome? What are the drivers of better patient care? So you could look into data, and data is again just like we talk about AI. Data is also everywhere, right? Like you have big data, you have small data, you have data crumbs. Data is everywhere, and there is a cycle, sort of a life cycle of data within from biology to therapeutics development to healthcare via data. And where things are going, so traditionally, we look at, maybe a study with, 100 patients and 100 controls. We look at their, just the measurements we could do in a hospital setting. But these days, we can not only do that, we can measure other things like genomics, proteomics, and other types of data. Data can, the medicine can be, traditionally centralized, like you have a hospital, you go there, you get that treatment. But these days, your doctor could sit, in the US, and then you could get treatment in India or within India itself in different parts of the country. You can still get the same type of care, and then even with technologies like 5G and beyond, we are expected that even you could do not just remote consultation, but remote surgery. We see that that type of demonstrations are coming. We can handle large scale data. We are moving from a GBs or TBs of data to petabytes of data, structured data to semi or unstructured data. And then we are also embracing more from a statistical approaches to we are moving towards machine or deep learning as well. And then when you do the studies, you could do studies that are with thousands of patients, right? Like it doesn't have to be hundreds of patients. You can handle that. And then one major aspect is that you can also, in a way, medicine is moving more towards a data-driven medicine, and rather than just the hypothesis-driven approaches as well. So this is where things are going, and there are a lot of ways we get data, right? Like because just like even the bioinformatics or translational bioinformatics or clinical informatics handle certain layers of data. As you can imagine, the data could come from a variety of sources. You could there is something called biobank. Biobanks are basically have a biorepository of a patient sample, but sometimes this will be linked with the electronic health record, right? Health record will have then their patient history of a patient, and that we can use to look into that. There are other aspects like disease registries, clinical trial databases, and then these days there is a huge growth of sensors and wearables that are out there that you can then use to connect even more data, not just the data that's available. So when you think about this, to study human biology or to do biomedical research, one of the major way we could do is to use electronic medical record. It's all about human biology. We capture real world data and evidence, and then this data can then be used for downstream analysis. But then why this is important? Because if you are studying human biology, what we currently do is if you are interested in studying, let's say, ovarian cancer, you create an ovarian cancer model, and in an animal model system, could be a mouse model, and then you try to treat them with the medicines that you are interested in, and then you look at that, and then you come and then you potentially go for after safety and efficacy studies, then go to human biology as well. But if you do have a large-scale database of human traits and disease state, you can do a lot more computing, and then you can predict or develop medicines in a much faster way. So that's the future. We're not there yet, but again, it's getting there. Almost all type of pharma companies now use this type of real-world data as well. So it's not just what's in what we can do with that, but then also there is a huge growth around digital biomarkers. So this is again, using other types of sensors and wearables. You can measure the cognitive states. You can measure heart rate, the environmental exposure we talked in the beginning. Patients can journal about their diet, and the walking, steps climbed, and vascular monitoring, muscle monitoring. So different types of things we can measure, and these are all important to basically look into different diseases as well. So this is again can collect a whole lot of data, so it's very, very easy and cheaper, and then a typical hospital visit associated with it. So now you've seen that there is a lot of data. This is about all of us, but there is also a lot of data. But then once you have a lot of data, what do? You potentially need to use some kind of a machine learning AI approach, right? Like that's what you could do. And what you have here is some examples. So these are all published examples. I'll be able to share the slides with you. And I just want to make sure. Okay, I think someone raised a hand. Will I take questions now, or will I take it?
No. Probably that's a mistake. All right, we'll take questions later. You can just
all right.
We'll take the questions.
Awesome, thank you. So as you can see here, we can use sort of group of machine learning approaches: random forest, support vector machines, convolutional neural network, and then use this type of approach to solve different problems. And this is all the work that's published. And as you can see here, I started my journey into machine learning and bioinformatics when I was a student with Dr. Ramanathan Sodamni at NCBS, and ma'am also have connections with Shastra University. We used to host students in the lab as trainees. So, and then also potentially have my friends have done PhD registration with Shastra. So I do have, I'm like sort of an academic cousin with Shastra. So this is where, for me, my journey into applying machine learning is started with this problem. Like, can you predict a structural feature or a structural phenomenon called 3D domain swapping from sequence information? And as you can imagine, we just heard that Demis Hassabis won a Nobel Prize for solving protein folding prediction using machine learning. So we attempted this problem. This was published in 2011, but we started working on this probably around 2006 or 2007, and into that problem as well. And it was one of the first approach, because I still remember going to conferences. We get, when we go to meetings and all, we get few people stop by. It was a niche area, but things have changed over the years, right? Like, as you can see that, we are now using this to look for safety in immuno-oncology trials. Like in cancer, these days, a lot of medicines or high efficacy medications are what we call as immuno-oncology medicines, right? Like, so for them, but at the same time, one of the key aspects is that they are combination drugs. So how do you combine them? How do we get a better safety profile. So we applied AI to look into that. We applied AI to look for latent descriptors of cancer in mammograms, because you can do mammogram, but once you connect that with the outcome, you can actually use mammogram and a machine learning algorithm to basically see, are there traces of breast cancer in that imaging that you could potentially look into. We applied it to a public health problem, right? Like, opioid crisis is a big issue in the US. People take pain medication, and then this pain medication become very addictive, and then they could overdose. So it could lead to substance abuse, mortality. So we used, again, the same group of these type of techniques to kind of look into, how we predict patients who might be at the highest likelihood of, going into some of this one as well. We used machine learning to develop a variety of approaches to improve what we call as drug repurposing or drug repositioning. So this is, again, as you can see, that this has been something that I've pursued throughout my career in collaboration with different labs around the world. And so this is just an application what AI could do. So you could do basic research level, you could go all the way to public health level. You could apply it's more or less the same algorithms, but then different applications. So now I'm inviting you all to kind of go with me, and the next 10-15 minutes or so, what we will do is we will primarily take. I'm just checking the time. We will primarily take you through an example of an artificial intelligence approach and how we can, how we typically develop this. And this is a lot of these materials are available online. Like I said, the publication link is on this deck, and I will share the deck with Dr. Suma as well. So you can start with defining a project goal. You can acquire a data set. The problem could be in any domain. Like I've shown you how we can apply this to everything from molecular biology to all the way to patient care, right? Like, and anything in between. And then you need to, once you get the data, you need to do data cleaning, data description, and then you need to build a model, and then you split the data into training, testing, validation. You do feature engineering, you select the algorithm, you optimize the model, you see whether you can generalize it, and then you look for certain metrics, and then you go to something like, you have a report, and then you can interpret that, and then you can describe it, make sure that they are reproducible, and then finally you can discuss limitations and alternatives. So if you are if you're interested in using any form of machine learning, this is how you handle your data, you perform your modeling, and then you will report this. So this has been pretty much the state of the art in the field for some time. And what happened? Things changed. And my attempt is to kind of introduce you all to that, what is that emerging themes in machine learning, and then we'll close by seeing some examples as well. But before we go, let's get this straight. So you do feature engineering. The feature engineering can, if you are an expert, or by working with an expert, you can select them. You can, if you have large scale data like genomics and others, you can do some dimensionality reduction. You could also do deep learning approaches like, autoencoders as well. As you go from here to here to here, a lot of this feature engineering has handled by the computer, by the AI system, than the subject matter expert. I like a bit of a hybrid approach, and I often try to do that as well. And then once you have this, you want to do the training and testing. So like, some of the key concepts involved, you can do what you call a bootstrapping, so, or a k-fold cross-validation. So the idea is that you divide your data into different Ks, and then, you kind of, each time you test your algorithm, you basically take each fold and then test that. Because if you are interested in predicting domain swapping, maybe you have proteins that are swapped, 10%, you have swapped 20%. So if I use, in my 50% or 60% of data set, if I have an enrichment for highly swapped proteins, I won't be able to predict the variability, right? Like, so we want to make sure that that's not the case. And it's extremely important that we do this type of approaches like k-fold cross-validation or bootstrapping and other methods as well. And then there are two major themes in machine learning: supervised learning. So, like I said, if you are interested in predicting domain swapping, you can collect a set of data, and then this was a work that we did during my graduate studies. So we had two interns from SASTRA visiting us, and they were actually part of a publication that we eventually wrote. So we had to collect data, right? Like which protein had swapping and which protein had, and then once you have that, you take the structure, you look into PDB, and then you need to annotate what is the extent of swapping. We came up with that concept, and then we created this beautiful data set, actually. So, and then we found at that time around 300 or so proteins with swapping, and then the rest of the PDB, out of that we selected R negative data set. So that's one approach, right? Like, and then these days we could even do an unsupervised. You could take the entire PDB. You can then look for these type of swapping in the data. It's from the data itself, right? Like, or are there, if you're interested in in a group of patients, let's take for example ovarian cancer. Are there patients with high high degree of tumor mutation burden versus low degree? You can actually do some kind of clustering as well. So these are two core themes in machine learning, and this kind of carry over even in a new generation modern machine learning approaches. Also this kind of carry over. So it's important to kind of get a gist of these two types of idea: your supervised machine learning and unsupervised machine learning, and then you can you kind of go from there to your solutioning. And before we close this section, so we can bring it all together. So there is a checklist created. This about the best practice strategy. You should remove, if you should check for any bias in your data set. You should always optimize your model parameters. And so in each and every step there are certain best practice suggestions are provided. My best practice suggestion is that, if you're working on a problem in biomedicine, see if AI can be applied, number one. Number two, give it a try anyway, and then see what's, if it's performing well. Because I've seen a lot of resistance that, is AI going to work? I don't have a big data. I only have a small data. It's okay, because these days, these approaches are very, very smart, and they can do things like one-shot learning, right? Like, with even a lot of companies are now developing algorithms to work with very small data sets. So the field is moving at a very, very exciting dynamic pace. So give it a shot. Like, I hope this portion was helpful to kind of think about that. But things have changed very, very dramatically. And then the truth be told, till 2017 or so, we were in what we call as an AI winter. So the machine learning, one of the algorithms that many of us use is random forest. It's developed around here, 55 to 61, and it kind of went into what is it called, an AI winter, and things were developing. Deep Blue was there, and there is this robotic dog from Sony. There is this thing called a vacuum cleaner that can work automatically, and this is one of the first thing I bought when I moved to US. It works really well. I'm sure this is all now available in India as well. We all have seen Siri, and then Alexa and others that came in as well. And then the Google Deep Brain did AlphaGo, which is again the same group that worked on AlphaFold as well, and that kind of changed. So so we had some algorithms coming from this era, and then and we started a few interesting things happening here, and then AlphaGo happened. And I wanted to kind of take you through that. What happened here? So few key themes, emerging themes. Neural network happened. Neural network, the classical neural network was always there, but then convolutional neural network happened. We started to think about new architecture, new style of developing machine learning models. So the example we were discussing, domain swapping, you needed to curate the data, you need to annotate the data, you need to then develop, your training and testings did. But then that all kind of changed. There is now pre-training, there is fine-tuning and transfer learning. And then some other emerging themes, including attention, embedding, encoders, and transformers that came. So as you can see here, so you have 2017 here. So you can see here, variational autoencoders happen, GANs happen. This is called generative adversarial network. We'll see them in a minute. A few transformers have introduced. So this kind of changed. And then you can see all the way here up to GPTs here. As you can see here, different type of algorithms. There is something called DALI happen. So GPTs around here. The first GPT is right here. So these are different approaches. So just like we were talking about support vector machines and random forests, this is the new era of AI. And these are even more powerful, right? Like, so they can do really, really amazing things. And, under the hood, if you look, if you go back to the architecture again, back to neural network, there are different types of architecture. We won't have time to see everything, but we'll try to touch some of these, like autoencoder, GAN, and attention network and all. But in my opinion, you can see examples of each and every one of these applied in biology. You could see that. And I want to share, let's say you're interested in using neural network to solve a problem, and this is the problem that I was referring to. What is under the hood? What is the architecture? What is the anatomy of that model, right? Like, so this is how we potentially design a model and adopting that to the use case that you have. In this case, we were interested in predicting latent expression of breast cancer in mammogram. So like that, you can and this is not a set architecture. You just like you step out of your street, you see different styles of home, right? Like they all have some basic things. They have a foundation, they have a wall, they have rooms, they have a floor, they have a roofing. Just like your architecture will draw it for you, and then the engineers and others masons will build it for you, right? Like similarly, under the machine learning approaches we use, this is the same type of an anatomy that you can see behind every algorithm, behind every application. So you might be interested in applying this to cell biology, maybe molecular biology, maybe in a medical use case. So you can use them. These are very, very powerful approaches. So in this case, we use them to look for early expression of cancer in mammogram data. So this is how we did our. These are the different steps that you could refer to as well when you design a deep learning model. But then, as I was telling you, things have changed. Like moving from the training, testing, validation paradigm, we now do pre-train. And what is pre-train? Train refers to the initial training of a model on a large, diverse set of data to learn general representation of the input data. And this is important, right? Like, so for example, I'll tell you, if you're studying cancer, you might have a lot of data on, depending on the country you're looking at, you might have a lot of data on breast cancer, maybe a little bit less data on ovarian, maybe even a little bit less on a subtype of cancer that's very aggressive called neuroendocrine prostate cancer. Right, like, but nowadays, if I'm interested in developing a machine learning model for neuroendocrine prostate cancer, the challenge was, back in the days, we would just have to collect all the data for neuroendocrine, then work around that. But these days, and interestingly, if you have other, even normal prostate cancer data, I can use that, or other subtypes of prostate cancer, I can then use that, pre-train a model, and then use that data to help with the downstream application. This really what we're doing here is we are pre-training, involved with training of a model or on a self-supervised or unsupervised task. If you're interested in looking at an imaging data, you can do that. The model learned to predict the missing or masked parts of the input data. So remember, this is what I was telling you. You don't need all the data ready to kind of apply this. You could potentially use a large model in your domain, and then you can do further from there as well. And you can do fine tuning. It's a process of taking a pre-trained model and train it into a specific data set. Maybe the first one you were looking at a marker one, but then the original model was built for that, but you could fine tune it in a way that you could have it predict impact of marker two as well. And then another theme is that called frontier models or a foundation model. And you might have heard about this in the news all the time, right? Like there is DeepSeek, there is O1, there is OpenAI, and there is other companies are building. Google has something called Gemini. So this is a pre-trained model that serves as the base for further development. These are a group of models called large language models as well. So I hope you get this idea that there is a paradigm shift in the way we do machine learning as well. And here's an example of how we can do that. You can definitely look into the paper as well. So you can do all of that, but you can also do transfer learning, right? Like, so in traditional machine learning, you might work on a single task, right? Like, your knowledge is not retained or accumulated. If, for example, if I'm interested, you can still use our domain swapping algorithm is available online, I hope. And then that's just one task, right? That it just does predict domain swapping only. It doesn't do anything else. But then again, it could do more than that. If we are architecting something like that, it can do more than that, right? Like, the transfer learning is again an emerging concept. You can have a data set one that will do the learning task one, and then output from that or the knowledge from that can go to the task two. So your data set and task one, data set two, task two, but that can be linked. But traditionally it was not like that. We were developing one model at a time. But that paradigm was also changed. So you have an example here in biomedicine, you can look at it. So it's a machine learning technique that allows knowledge gain from one task can be applied to another one as well. So that's transfer learning. And then there's another concept, comes embedding. Embedding is like I was telling briefly about machine learning approaches using electronic health record data. So if you have access to patient data, let's say in Tanjore side, if you have a hospital and if SASTRA can make some agreement to access that data in a safe and secure way, you can get data on patients, and then that data can be very powerful to come up with strategies to improve healthcare. But then computing on that large scale data is really, really challenging. You need access to GPU and all of that type of approaches. But then to do fast computing, you can do some kind of embedding. So what we try to do here is that the main idea, you can take multi-dimensional data or high dimensional data. In an EHR, you will have information about the disease, medication, lab tests itself, thousands of lab tests, procedures. You have everything from a simple surgery for a broken hand to all the way to complex cardiac surgery. Like, these are all procedures. So each one have different type of data captured in its duration. So what we could do is we can capture that type of complex datascape and then use them for downstream application. So you can embed with words, sentence, documents, images, graphs, knowledge graphs. So all these type of things are possible. So here, really, what we actually use this technique to kind of do, what we call as disease cohort identifications. This was a project done when I was in New York. So with the large health system called Mount Sinai Health System. So we applied a technique called word2vec, and then we created this embedding concept from that data. Then we use these embedding vectors to look for patient similarity. And so now you can search, like, literally, hey, is patient A similar to this versus B? Not just by looking at one type of data, but by looking at the entire type of data from that patient. So you can start from one patient and then look for the very similar patients and cohort them. So that's just one application. So similar to this, another concept that's coming and what your machine learning algorithms can do is encoding. So I might ask you one question if one or two members can respond to me. So this is Bensi, what would this be? Just as a question. And I want to make sure you're all listening. Can someone answer? What does that word comes to you? Anything. What do you think when you hear a word like that? You can type it in. It's okay. We can.
The audience can type in the chat.
Okay, someone said a drug is come to the mind. Okay, very good. I need one more answer. It's a name. Excellent. Okay. So what this shows, if you can take a look at the chat, Pranavadiyani said that this is a drug, and whereas Shashikala says this is a name. Right? Like, now I'm going to give you a little bit more context. Now, what do you think? So I'm giving you a little bit more information. 1-year-old from Maryland, likes to play with water, friendly with kids, comfortable with other pets, unconditional hugs. And I need one more answer. It's a pet. So excellent. I think Shashikala was thinking in the right direction. It's a dog. So just now your brain is, what happened right now is a beautiful neurological phenomenon, neuroscience phenomenon. So initially I just showed you a low-level information. I gave you a few more features, and then we came to this one. So this is now brain is encoding it a little differently. This is an important part. Now just imagine you can, if you're working on a large-scale problem, and your data can be encoded in a lot of different ways into your approaches, algorithms. You can compute very fast. You can infer, build models very fast. And this is something we use all the time, right? So it's a process of representing data in a specific format. We takes the data, transform it from the original data was a dog, that was a pet, right? Like, but I can just represent it using a name, any of these features, or using a picture. Right, the original data can be represented in a lot of different encoding format, and you can do this like one-hot encoding. So BENZI is just a one-hot encoding. You can label them binary, ordinal, hash, targets. There are a lot of different ways you can do that as well. So this is now your algorithms can do this now. So not just embedding and not just you can do also encoding. And then this is another interesting concept. I need at least 2 more answers, but I'll tell you. Imagine I know this evening in India, but imagine, it's morning and then you want to go to the campus, right? And it's 8 o'clock and you're going to the campus. What would be in your mind? 2, 3 answers very quickly. Okay, we got one. Someone else? Weather. So yeah, we all think about weather. We'll check that, right? What else? Traffic. Excellent. That's great. Quickest route, time. Super. This is excellent. So, but irrespective of all of this, go early. I get it. Irrespective of all of this, your attention is on one task to get to your campus. Right? There are a lot of other things that can happen, but in the end, your ultimate aim is to get to your campus as early as possible. So what happened in computer science is that this is extremely important that you get this concept. Your machine learning algorithms can now do this. What does it mean? It can do attention. And this very exciting paper that came, it's called "Attention is All You Need," And what happened is that it's a mechanism that enables models to focus on specific parts of input data while performing a task. It mimics the selective attention mechanism observed in human cognition. We prioritize certain information over others. Now let's go back to our responses. It's the weather, traffic, quickest route, time, go early, bye. All of these are relevant, right? But our ultimate aim is to go into, because we're not going to check weather all the time, right? Okay, it looks sunny, it's good, let's go. If it's raining, let's get an umbrella and let's go. And then if there's a traffic, but should I walk? Should I take the bus? Should I drive or my bike? These are other questions around that. So your human brain processes a lot of information and prioritize certain points over the others. And your machine learning algorithms can do this as well. So So that is but remember, like, that was 2017. This approach we came out. So that new. And then now comes sort of this section's last topic is transformers. The transformers refers to a type of neural network architecture that gained popularity. But what it does is basically, I hope many of you use GPT algorithms and Chat GPT and others. And then again, it employs a mechanism called what's under the hood of this? It's attention. And you've seen attention before. Okay, so the beauty of ChatGPT, when you ask a question to ChatGPT, it's giving you an answer, and it's kind of almost like magic. It's because it's looking at that one task, and it's looking at all the information, but it's looking at that one task. And again, these things can make errors, but you can always refine them, improve them. But they have achieved remarkable success in natural language processing. And then one example here is that translation is happening in some application that you have linked to the talk today. So similarly, these things are at the core of this. So I hope this was useful. So we kind of took a quick tour through some of the machine learning concept. And similarly, we also have other emerging concepts called generative adversarial network. This basically can help to, if you have a study, you only have 30 samples, but you can generate results, right? Like you can simulate them, and then again use that to improve your study. So this is one of the approaches. And then an interesting example is that these individuals are, let's say they are real. What we do is we start from the source, and then we apply to this one, right? Like we take these sources, and then this is the destination. So let's say if I combine this gentleman here and this curve here, this is probably the one I will get. And again, this is just one representation. So you can imagine you want to do a transcriptome generation, you only have 50, but the other 50 you could generate. We are not doing that extensively yet, but I see that happening. Like a lot of times we are now simulating data before we go into do the experiment. So this is also another theme that we see as well. And then again, to kind of conclude on that session, so language models, which we all use now, they are transformers, right? Like, and then there are different types of transformers, BERT, OpenAI, and others. And these are large models, like the parameters are in millions and billions, and even could hit trillions as well as we go. And it's very expensive to build these ones. It costs, some of them initial ones cost, very high, very much into millions of dollars to create these type of models. And then again, if you are interested in looking at going further, like what is in a GPT, most of the concepts we saw today, right? Like it has a transformer encoder, it has pre-training, it has a decoder, it has an attention mechanism, and professional encoding and fine tuning. So this is the concept that's happening here. And that's that. So if you say, starting with my dog is cute, he likes to play, because then it looks into a lot of different types of encoding, and then based on that, it kind of come up with that result. Basically, it looks into the first word and then try to look for, it takes the data, decode the data, and then use the attention mechanism and do some personal encoding. This is what really happening under the hood. So I hope all of you appreciate this kind of growth that machine intelligence is having, and we could apply them to our problems as well. So I just want to check how much time do I have now? Maybe 10 more minutes?
Sure. Yeah, you can have one.
Yeah. Okay. So the idea is then to use this type of approach. There are a variety of ways we could do machine learning, right? Like and one theme we will see today is called a graph machine learning. And the idea is to quickly go through 3 examples, and and here one is to look for a target discovery, because it's at the core of developing therapies. I will also discuss briefly about looking into clinical trial data, and this is an exciting area, and I don't see a lot of work coming from India, but you could all do this. And this is again, India does a lot of clinical trial, and a lot of data is available, but we need to really ask more questions with that. And then I'll introduce a concept of diseaseome and prescriptome as well. And what these do is, this is more towards the target drug discovery. This is a concept called clinical trial optimization, and finally, population health analytics. So in this case, what we are interested in using a graph-based approach to look for targets. And ovarian cancer is a challenging cancer. It is there are some therapies, but again, one of the interesting part about therapies is that maybe even for the best drug, 30 to 40% is the maximum percentage of patients respond to that therapy. So you definitely need new therapies so that patients get better outcomes. So in this case, what we did was we looked at a protein interaction data. So there are different ways to look into these graphs. But we did an approach called, what if we look, the concept of the neighborhood search, right? Like, so, network expansion is an approach that can help in improving function assignment, pathway assignments, predicting members of protein complexes, building disease networks. So we kind of thought of, what if we apply this to ovarian cancer? We start with a very well-known, highly curated seed network of ovarian cancer genes. This is the most lethal gynecology cancer. There is a lot of drug sensitivity, and drug resistance is a major challenge. Patients could resist and won't stop responding to therapies as well. So we definitely need new therapies. So how do we do that? We need new targets, right? Then only you can develop that. So we came up with this. We used this approach, network expansion. We applied it to a high-quality seed network, and then used that network to extend it further. So we started with, let's say, give or take, 72, and then we added another 20 high probability genes to that one, and then we compared their, in terms of their interactions, co-expression. I think this is the seed, and then this is the expanded one. So they were capturing more, for example, more pathways, more interactions, more expression. So some of the genes we added might be an interesting target. And then what we did, we looked at the network and computed a metric called radiality. And then, as you can see, and we used one case and one control. So June is a very well-known ovarian cancer. So June is coming right up here, but we got a bunch of genes that's ranked higher than that. And then we also have, we also added a negative control to that as well. So this was CDC14B, which has a role in cancer in general, but not in ovarian cancer. And we found this, and this is a coil protein. And based on the analytics that we did, we were able to look into the structure, and then we did not stop there. We wanted to make sure that this is the right target. So we did some downstream studies, right? Like these hub properties of that network. Do we capture that in an experimental setting? So we tested KRCC1, June, CDC14B genes and started silencing them in a cancer model. And then we also did additional studies to inhibit them. And inhibiting them, results in DNA damage and apoptosis. And as many of you may know, apoptosis is one way we can kill the cancer cells, right? Like, so if you delete this, you're seeing an output like apoptosis as well. And so we did the cell line studies, it worked, and then we went into looking at it from an actual animal model, right? Like, so what we found there was, if you silence KRCC1, it inhibits the tumor growth in mice. So we were able to kind of come to a target, using a systems biology approach for ovarian cancer. And then, now additional work is going on how we could potentially target. It's not an easy target because it's a coil protein, so it's not direct approach. It's sometimes difficult. So, but irrespective of that, we want to share this as an example here. So that's one way we do that. Now, let's say, you identified a target, you developed a drug, and the drug is, in going through clinical trial. And clinical trial is a very high effort, high investment activity, and then a lot of trials fail. You might have heard about this, 80-90% of the trials fail. And there is reasons, there is operational reasons for that, and then also the medicine is not working. That's again the reason trials fail as well. And one of the major challenge with the clinical trial is enrolling patients, right? Like, you can enroll patients based on your protocol, but they may not stay. due to various reasons, patients drop out. There is legitimate reason as well as illegitimate reason. So we are interested in this. Every pharma company is interested in this problem actually, like how we can improve our patient enrollment, life cycle or clinical trial enrollment life cycle. So we applied, AI approaches, the same type of approaches, trying to see how we can predict patients who are more likely to drop out. And again, one of the reason is that side effects is one reason, and then there is also other types of reasons as well. So what we did was, if we could predict side effects of clinical trials a little bit better, we could potentially address this, right? Like, if you are to go study this particular molecule in this clinic, in this disease setting, probably we could do that. So we applied an approach called a graph machine learning. So last time you saw more of a network analytics sort of approach, but here the interesting part is, if you have a large graph of disease, drugs, and side effect, you may have it represented for 1,000 drugs, but now you can use that information to predict side effects for a new drug. You can use that graph to do what we call as a link prediction. Once you have that data. So we build a large scale workflow for this, and what we showed here was, I'm not getting into all the technical aspect, but we build a model that's graph based and then also an array based model, very traditional Excel based model as well. Then we showed that the graph based model was able to predict side effects a little bit better than and I'm talking about the magnitude depending on the disease, anywhere from 1 to 5% improvement, which is significant in this space actually. So we used a bunch of different type of approaches here. These are all sort of algorithms available to predict outcomes from a graph, and then again our sort of structured approach here as well. So we were able to do that, and we created this trial level side effect estimation. So if you have let's say this is the network representation of the data. So you have an adverse event respiratory failure which presents in this clinical trial, and this is for this particular disease or autoimmune, and then again so on and so forth it can go. We were able to predict that, as in this graph you can see two types of analysis with the graph, how well they're predicting, as well as the array. So as you can see here, graph was able to predict really better than the array-based approach. So again, just an example of what we could do with this. And again, this is an area that's, in my personal opinion, we do not look into a lot of this reanalysis of clinical trial data. It's an exciting area. And this also is a publication available, and you should be able to look at that. And so you've seen one example on target ID, you've seen another example on trials. Now we are going to population scale. So what if you have access to, remember I was telling you, like, you have access to all the patient data from your city. So in a similar way, we had access in the past, from New York City. When I was at Mount Sinai, we had access to around 1 million patients or so. So we looked at something called disease trajectory. This is a very interesting concept called disease sequelae. So what the basic notion is that, can you predict the next disease, right? Like, and this problem was addressed a while ago. If we're doing it now, maybe we'll use language models and others or transformers, but we did it in a slightly different way back when we tried to solve it. So what we're trying to do here is we are trying to create disease networks from real world data, but then asking the question, based on sort of the self-reported ancestry, are their disease network different or are they same compared? So here is what is if we base this to European Americans, for African Americans and Hispanic Latino, what happens with their risk profile? Some of them are higher, some of them are lower, and how we can use that. So that was the kind of concept. So we start with the disease X, then go to the Y, and then go to the Y to the Z. So you can basically, by looking at records, you can measure this in a you can create these disease sequences, and then we created separate networks of patients as well. So what we did was, we created trajectories for approximately 2,000 disease using 1 million patients as well. And here, if you can look at any of the disease of interest, I would say focus on your red, one of the red that and then just remember their neighbors. But this is for European Americans. But if I go to the next one, you see that some of the neighbors change. That means the trajectories are a little bit different. And then when I go to African American again, that changes as well. So this is important, right? Like, so let's say if you have a patients coming into the hospital and the doctors have access to this type of disease equilibrium prediction, that can definitely, we can then help the patient with other types of approaches, right? Like that's a possibility as well. So we were able to build that. We were able to find ancestry specific disease nodes or trajectories as well. And then this is now can be used as a predictive model. And then you can do this with medication data in a similar way. And you can also predict what would be the next medicine a patient could get based on the data as well. And we created a massive direct pair sequences as a graph, and then this graph can now answer different questions as well. So what I want to do is, these are some of the ways we could look into these type of large scale data. And again, this could also address another public health challenge called polypharmacy. If you have a grandparents, or even if you have, you can talk to your parents, and they might be using different medications, and this often leads to primary and secondary side effects and poor outcome. This kind of trends you can measure from these type of approaches as well. So even though if it's a basic level question, you could bring it in the context of public health as well. So I want to stop here, and then this is 3 examples that we discussed, but India could do a lot more, right? Like, with the kind of algorithms, approaches, and methods that we have available, there's a huge opportunity here. There is funding priorities needed, but there are things that are needed, but there are things you could do as well. And then again, in terms of education as well. So I advise students from a program in the US called NMed that combines engineering and medicine, like that type of programs, and students that does MD and PhD, means they're MBBS and PhDs. So there is a lot of ways, and then of course, computational biology, bioinformatics groups can get access to data, right? Like the EHR data. All of this was possible because there was a safe and secure way to access data, right? Like, so students can then start working on that. But then again, think about it, scaling scientific evidence-based and optimized healthcare to 1.2 billion people is an immense opportunity. And I invite all of you to think about that. And, before I close, the data availability is growing. Data is measured, captured, and then you will see more AI approaches than ever as we go into next decade. And there will be, we all hope that there will be a lot of standardization, because since this is still in early stage, a lot of these regulatory aspects or other laws and other rules and regulations are still being developed. We will hopefully see in at least in your lifetime, you'll all see AGI, and what that stands for, general intelligence, right? Like, instead of and that's where this is going. I hope you got the gist of that, going from a custom built model to large language models, and then this is where things are going. And I hope at least one of you are excited to kind of look into some of these problems, and then I hope we spend your Saturday evening and my Saturday morning. It was all worth. And none of this is possible to do, without access to a great team, great colleagues and co-workers and mentors. I want to thank them all. And with that, I will stop, and I'm happy to take questions.
We are open for questions. The audience can type their questions, and I think already we have a question from Sasikala. Chat box.
Okay, if somebody can, since we are using AI in clinical, does it align with ethical guidelines? A short answer is yes, but if you are applying in a live trial, you need to get it approved by your regulatory agency. Is there any drug that is AI-driven design which comes to clinical trials? There are multiple. That's a question. That's a very interesting question, and I would say there are. it depends on the answer is, short answer is yes. Long answer is every drug that you see, that came out, that comes out these days have some level of AI use that. Can we combine simulated data set to real data set and do ML since the patient data is too small? Yes, and that is one of the point that I was making. You can take a a foundation model, and then even for your disease, if the data is small, that's not a problem that you can address. So these are great questions, and I will send the list of drugs separately. So there are some articles that I can share. Okay. If someone can read the questions, I can answer them if that's okay.
Sure. I would read that. So there is
Thank
another
you.
question from Priya Vishwanathan. The question is, can we use transcriptomics data deposited in the repository for biomarker analysis? Is there any parameter that needs to be considered when we are using it?
Yes. So Geo is a gold mine, right? Like it's called Gene Expression Omnibus, right? That's the one even we use, because even we are a pharmaceutical company. We have access to good funds and to support our research and all, but a lot of time is important. A lot of time that it takes time to generate data, right? Like, so if you're interested in, let's say the same thing, right? Like, if you're interested in a particular cancer, there might be, 10, 20 studies already available in Geo. And we often take that, and then on top of that, we do our analysis. So I would say, curate them properly, read about the experiment that's been done, and then use that. So that's what I would do that.
So the next question is, how do you see the future of clinical AI in terms of ethics and explainability?
So the ethics, great question. So what happens is that you have to think about it, right, in a slightly different way, right? Like, let's say we apply AI in a clinical setting. We wanted to make sure that it's validated, it works for the target patients. It doesn't do the model doesn't have any bias or discrimination. You want to make sure that there is a governance body, understand what is happening with that. So what happens is that if from the hospital that's deploying that, there should be an AI governance committee that look into this, and then also whether it's a state or country level. So that's where things are going, because I'll tell you one example, right? A simple example. That's not a simple, very important example. So let's say you did an MRI today, and you need to interpret that, and what happens is that if the condition, and I can tell you, we worked on this disease called hypertrophic cardiomyopathy, and what happens is that, let's say if you are someone who's training to be an athlete, your heart muscles get stiff, right? That's a phenotype that you get. Similarly, if you do that, if someone has an hypertrophic cardiomyopathy disease condition, their heart muscles also become stiff. And then what happens is that sometimes patient who might be an athlete, who might be at high risk of this, could come to your clinic. And how do you differentiate between the two? So it's extremely difficult to do that actually in reality, unless you are someone who has seen hundreds of patients that are athletes, hundreds of patients that are with HCM. It's very difficult to kind of diagnose that, and you have to send it to your network, and it takes days to come back with the result. But you can design an AI model to actually differentiate between the two. It works really well, because AI can not only look at the traditional parameters, but other elements within the image that we don't capture with the human cognition or human eye. But then the challenge is whether this is acceptable as a medical grade diagnosis tool. Have we tested it properly? So in that way, would you rather use an AI and diagnose it quickly, or would you wait it out? Because HCM is a very significantly serious condition, right? Like, so this is the thing, and then that's the part of the ethics, and then come to explainability. In one way, what is AI thinking, right? Like, even when we were trying the Benzi experiment, or we were discussing the attention experiment, we were looking at each one of us had different answer. You might have seen that, right? Like, so AI will look for all of them or none of them, right? Like, so that's the thing, right? Like, so the older approach of going with training, testing, set features, that's changing because these are becoming smarter. But now what we do is you can apply some kind of explainability to the algorithms and then derive what's the features driving this prediction and how likelihood this is, in likelihood this is an error versus an actual observation. So you have very advanced methods available to address that, and that is actually available to address the explainability part. Both are important, but we will be in a future where AI will be used, whether we like it or not. It's coming, and I would encourage, these are great questions, some of you to think about being that torchbearers of that field, because those who have the background of bioinformatics can do a PhD in this area and then drive this for India and beyond. That's an excellent area. You need understanding of both. Yeah, I think we have one more question.
We have one more question. Please brief on the graph-based model, like how we have to prepare the data, what are all the associated parameters that need to be considered on prior.
Okay, so the interesting part about graph is your typical data comes in an Excel sheet, but from that Excel sheet, each row, each column that you have, you can think of it as a node in your graph, right? Like, and then if you have relationship between the nodes, you can derive edges. So graph has basically two components, right? You have a node, you have an edge. You have another node, you could have edges, and so on and so forth, right? So that's something that's how you can do that. And if you're interested in biomedicine, like you get PPI data, they're basically 2-column data. But a lot of the data that we saw today, we created the graph. We created the knowledge graph based on the knowledge in the domain, in that subject that we're talking about. So I would encourage you to think about the problem. If you have an Excel sheet, from that you can create that one. And then graphs are working. So some of the examples I showed today, these are examples where graphs working, but there are areas where you don't need a graph, right? Like, the other approaches could work. But I would highly encourage, because of the linked nature of our data, to try it, because what I saw in a lot of the experiments we shared today, graphs perform better than the traditional approach.
Maybe we have one last question, sir. So the question is, how accessible is healthcare data for research purpose?
So there are, I don't have an exact answer for the Indian resources, but I can tell you there is something called MIMIC. This is an ICU level data from hospitals in Boston, and they provide de-identified data for all the patients that are hospitalized in an emergency ICU, like our ICUs. So there are other projects like MIMIC that you can get access to data as well, and you will be able to, and basic premise is that you need to prepare a research abstract and then you share it, and they'll approve that. And that's, MIMIC is just one example. But if let's say you're interested in genomic data and clinical data, right? You can access that from UK Biobank. They provide you access as well, but it has to be through a supervisor, or you apply, then supervisor needs to approve that as well. So that's how they work. So there are a lot of data sets available, and I think there might be data sets available from India as well. Yeah.
Okay. There are a couple of questions about, is there any book that you could suggest where they could learn from very basic things, or any study material?
So what we saw today, it's an interesting question. I create a lot of these slides to kind of share a lot of this emerging approach. So I've not really seen one book that cover all of them, but I would encourage you to participate in discussions online, right? Like, there is something called, I don't know if you use BioStar. It's an online discussion forum. what you call, there are discussions happening in other sources like LinkedIn and others as well. So I don't have a book recommendation right now, but I can probably look it up and send you. But I would say the one way is to have have a study group in the university, right? That talks about some of these things, right? Like, and then pick papers and learn, right? Like, this is the best time of your life, I should say, right? Like to experience and learn and try and tinker. Download that data, build that model, get your hands dirty. Of course, books and concepts matter, but I would say look at it from a more practical perspective. Yeah, that's my suggestion. And you have a lot of online materials as well. So I would say one resource you should all look into is something called Hugging Face. Yeah, exactly. The name is type it in Hugging. I think it's huggingface.com. So this is a repository of machine learning, like the models we were talking about. This is a good place, and yeah, very, very useful for looking at things we discussed.
Yes, sir. Thank you. Now I call upon Ms. Lasya for proposing the vote of thanks.
All right, thank you.
Good evening, sir, and everyone. It's a privilege to deliver the word of thanks for today's insightful webinar on emerging applications of machine intelligence in therapeutics development and healthcare. First and foremost, a heartfelt thank you to our esteemed speaker, Dr. Khader, for sharing his expertise and invaluable insights. The deep knowledge and thought-provoking perspectives have truly enriched us, sir. I would like to extend my sincere gratitude to Shastra University management as well as the dean, associate deans, and professors of CVT of their unwavering support in making this event possible. A big thanks to you, to the organizing team, for the seamless coordination and efforts in ensuring a smooth and engaging session. And of course, a special thanks to all participants who enthusiastically and active engagement making this discussion more meaningful. We look forward to more such enlightening conversations in the future. Thank you.
Thank you, and pleasure is mine. And I really appreciate the students staying throughout the talk and great questions.
Yes, Shamir, thank you so much. So we have been trying to arrange your session for a long time, maybe even before COVID itself. No?
Yeah, so
it's a great privilege you are here, and it's a great session, sir. So thank you for very much for the simple explanations about the attention transformer mechanism, all these details. It will be very useful for the students.
All right, glad to hear that.
Yeah, so one more like maybe question, like the majority of the participants are students. So about their how they have to plan their career to be placed in such companies, how to build their AI model for their skill set.
Yeah, so again, this is a very sort of a transformative time, right? Like everybody, on one side, it's a great place to be a student, right? Like, and then if I'm again in an undergrad or straight out of high school, I would still study. I was telling my daughter, she's not, she's in high school, but in the US, people think about this very early. I would recommend to study computational biology and bioinformatics because you get best of both worlds. So that's one thing. And then what's changed with all these things like language model, access to data and code and everything, right? Like, yesterday I was doing I wanted to do a Google search, about, collecting a specific information. And then I asked my daughter to do that. She asked one of these GPTs, okay, create a table with this information. And I was telling her, I used to take 45 minutes to collect that information. And we were in a car, we were driving, and she got that in a span of maybe give or take 20 seconds. So we are in we always talk about this information era, this, and then it was primarily, but we were doing the search, the user. So now you have each and everyone in this group have an assistant. Think about that.
Yes.
So this is a transformative period to be a learner, to be a student, and use it to solve problems like this. Now the why and the how and the what, right? Like you get a lot of help to do that. So think about a problem you are passionate about. It could be as tall order as, I want to find a medicine for this drug, or I want to improve the healthcare of this, my community. You can make a difference. These are mere tools, but in the end, the student is the one that's driving. You have all of this information. You have access to this computing. You have access to other resources. Or if you are, planning to do PhD, think about how fast and how optimally you can do your work, right? Like, because I was telling about the same thing, right? Like when we were doing our domain swapping research, I literally had students reading the paper, look for swapping, and you can now do that with I'm not saying 100% using a natural language processing, but maybe 80%. So we could, I could have published that, 6 months before, right? Like, so think about it like this way, whether your skills in data, AI, computing, and machine learning, there is a lot of tools, a lot of ways to do this, right? Like, that has increased. Earlier, back in the days when we were doing the biggest supercomputer was in, one of them was in Pune, and then again, luckily we had access through NCBS, so computing was not an issue. But the software, there was a lot of people were using just the MATLAB. Now this has changed. So if you go to the Hugging Face website, you see that where the models are available. All the models are available. There's plenty that you could do, right? Like, there is a lot that you could try, and bring, publish them, and then learn from them, and then improve them as well. So there is a lot that you could do, I would assume, and in a much faster pace. But think about problems. Think about what you are excited about. It might be creating a new variety of a crop in your region. It could be that you can, with the kind of things we talked about. I know today we focused on biomedicine, but a lot of this you can apply in agriculture. A lot of this you can apply in developing better policy, better resource allocation, so on and so forth, right? Like, so these are mere tools, but in the end, it's the students, the research community, the research labs are the drivers, right? Like, so like I said, I'm trained. I did my master's in India at Mahatma Gandhi University. Then, I was fortunate to go to NCBS at that time. I went there for a conference, actually, and that's how I learned about Professor Sudhamni, about her research, and I went back, as a research assistant, and then became a student. So each and every one of us, the path would be different, but it's about the goal. My goal was I wanted to go to NCBS for my PhD. If I'm doing a PhD, I'm doing it there. Otherwise, I'm not doing it. That was my take on it. So like that, right? Like, and I had friends who went outside, who went abroad, who went to other places. And then what I realized was the training I received in India was as good or sometimes even better than other places in the world. So what you have, if you might think, okay, maybe, I should get trained somewhere else. I've been through that too. But I can tell you that I think I did okay eventually. But the training, the teaching, the focus on doing things and the persistence, that matters. And those are, if you ask me what, when I hire somebody, I hire someone who has tried a lot, who's persistent. Others are soft skills. You might know maybe a little bit more Python, the other person know a little bit less Python. Does it matter? It doesn't matter. You have GPTs to help you. So, the core aspect is, how much trainable are you? How much you can learn new things? And then what problem that you're passionate about, and how much that you are interested in spending time on. Now everything is available online. If I ask you, create a machine learning model, and this is one challenge I'll give you. 3D domain swapping data is available online. Find it, create a model. and send it to me and maybe Professor Damini as an experiment. As an experiment. I wouldn't give any other instruction. I'll tell you there is sequence data available, and with the tag of positive and negative data set, it's available publicly. Right? Like, so just as an example, but like that, think about, look into your community, look into your own personal experience or things that excites you, and use AI as a tool. Don't think AI will take your job. AI is not going to take your job. AI will take the job of those who don't embrace AI. Because in the future, just like we say, right, like what is the core skills you need, right? If you ask me, if I'm hiring somebody, the core skills, the number one skill I would require is communications, not data science. Even if I'm hiring a data scientist, the major part is, the number one part is communication. What's your emotional intelligence? How do you handle a room full of people, right, that may or may not agree with you? Because, whether you go into research, whether you go into private or, public service, not everybody is an expert in AI, right? Like, and people understand this a little bit differently. So your job is to kind of bring those group together and then, say that this is about it. This is the impact we're expecting, right? That needs, that clarity in communication, right? Like, and then again, your math, your computing, these are skills that, nowadays it's learning. It is very easy. If you think there is a gap in your, sort of learning arc, it's very easy to address that. It was not like this. And I'm not that old as well, right? Like, I lived through the internet era, right? Like, and now is the best time. Like I said, I took 45 minutes to create a table. My daughter created that in 20 seconds. That's just an example. So I'm sure all of you will do great things. You should all do great things. And AI is your friend. It's not an enemy. It's not going to take your job away, or, but get your hands dirty, right? Like, get a data set, apply it, let it fail. It's fine. We'll fix it. That should be the approach to learning. That, that openness to learning matters a lot, right? Like, so that's the major part, right? Like, I'm sure being a student could be challenging these days because they're saying, okay, I will take away. This narrative was always there, right? Like, when I was growing up, it was computers taking all the jobs away. And then what happened? Right? We have all seen that, right? Computers are there and humans are there too. So in this round, it will be like that. The hybrid is the future, but maybe a little bit more machine. Machines will be a little bit more intelligent. But you still need doctors. You still need to, if you need to take your blood, you still need to analyze them. There is a lot of things you automate, but you still need experts, including computational biology. And I'll tell you, I'll stop this with, when I read about bioinformatics in an article that's written by someone. I'm originally from Kerala. His name is B.S. Warrior. He wrote an article. I read it when I was in my undergrad. And I liked that, right? I loved medicine also. I also like many other things. But I already became undergrad, then you can't go back to medicine, right? So then you can use computers and this one. That was really exciting. And I can tell you, I never had a day without excitement in my career. Of course, there's pressure of life and everything, but in terms of, I get to do a lot of new things, and that's the joy of working in an AI hybrid area like computational biology and bioinformatics. So, like, I've shown you the kind of examples we can apply this approaches thinking to everything from basic drug discovery all the way to large scale patient analytics. I was trained like you, like in a university in India. I did a PhD in India, and I came to postdoc here. But the training was solid. I should say that I have the retrospectively I realized that I had the best training, right? Like, so get your hands dirty. That's my only advice. Go find the data set, do it. And this is the best thing to study now, right? Like, I don't want to prescribe career path, but the jobs are in hospitals. Every hospital will have a omics lab in the future, right? Who will handle all the data? Every pharma companies have huge computational biology roles. So just think about it, how many pharma companies we have. And in India, a lot of them are still manufacturing focused. They could be moving to R&D focus too. Many of them are now, but there will be growth there. The clinical trial, like we were discussing about that, right? Like, there's a lot of back offices for clinical research organizations, CROs in India. And then biotech companies, for other non-human life science companies, veterinary companies, they all need this type of expertise. So I'm not prescribing a career path, but it's about what you want to do. There will be a job for each and every one of you, if you, that's the thing, or the research position that you're interested in. So I would say it's the best time to be a student, and even best time to be studying all of this, because this is a unique opportunity. You can give back to community and the world as well. Yeah, it takes time, but you need patience and persistence. But I'm sure Dr. Suma and others are teaching you that. But that's an important virtue, right? Like a drug discovery project takes 10 years, maybe 20 departments, and $2 billion in expense. Right? Like every molecule that I work on now will eventually become a medicine in 10 years. So that's how things works. And we are trying to reduce the timeline, but this is going to take time. So that matters. Even with all the AI that we can throw in, how much it's going to save time. So I want all of you to come into the field and reduce it to like 2 years. So we have medicines in like that, time scale like that. So I hope that's helpful. I don't know, that was the right answer.
Yeah,
but
it was a great, inspiring message. Great one.
Okay,
Yeah,
thank you.
Good, for the students to take take forward their career in the computational biology field.
Yeah.
And we look forward for such direct session maybe in future.
Sure. Yeah. I'm
Let's
happy
hope
to
for
help.
that.
Yeah. All right. Thank you,
Thank
everyone.
you.
Have a lovely evening. Thank you.
Thank you very much, everyone.
Yeah.